Hello dear students. Today we will discuss an important topic which is techniques of plant tissue culture. Plant tissue culture is a technique with which plant cells, tissues or organs are grown on an artificial static or liquid nutrient medium under aseptic and controlled conditions. Plant tissue culture helps in development of whole plant from a single cell, tissue or organ using various techniques. This genetic potential of a plant cell to produce entire plant is known as totipotency. In this lecture, following steps of plant tissue culture will be discussed: washing of glassware, preparation of nutrient media, selection and surface sterilization of the explants, inoculation, incubation, regeneration of plants from cultured plant tissues, hardening or acclimatization, and transfer of plants to the field conditions. Let us discuss the first important step of plant tissue culture technique, which is washing of glassware. All the glassware that is used in the process of plant tissue culture is soaked in detergent solution like laboline for a suitable time duration, preferably overnight. Then the glassware is rinsed, first with tap water, followed by washing with double distilled water. After washing the glassware, it is placed in trays. Finally, the glassware is dried in an oven at a temperature of 75 degrees Celsius. The second important step of plant tissue culture technique is preparation of nutrient medium. The tissue culture medium is composed of following components: macroelements. Macroelements are those substances that are used in large quantities. They include nitrogen, potassium, phosphorus, sulfur, calcium, and magnesium. Microelements. These are those elements that are used in trace amounts, and they include manganese, iodine, copper, cobalt, molybdenum, boron, iron, and zinc. Organic supplements. Organic supplements include vitamins and amino acids. Among the vitamins, vitamin B1, B3, B6, and myoinositol are used in the plant tissue culture medium. Among the amino acids, glycine is most commonly used. The most commonly used carbon source in plant tissue culture medium is sucrose. However, carbohydrates like glucose, sorbitol, or maltose are also used in some preparations. Gelling agent. The most common gelling agent used in plant tissue culture medium is agar. Agar is derived from red algae, Gelidium amanaceae. The less commonly used gelling agents in plant tissue culture medium include gelrite and agarose. Plant growth regulators used in plant tissue culture medium are auxins. In plant tissue culture, auxins promote cell growth and cell division. The most commonly used auxins in plant tissue culture are 2,4-dichlorophenoxyacetic acid, naphthalene acetic acid, naphthoxyacetic acid, indole 3-acetic acid, and indole 3-butyric acid. Cytokinins. In plant tissue culture, cytokinins help in promotion of cell division and differentiation of adventitious shoots. Some of the commonly used cytokinins in plant tissue culture are benzylaminopurine, kinetin, isopentenyl adenine, thidiazuron, and zeatin. Gibberellins. In plant tissue culture, gibberellins help in cell elongation. The most commonly used gibberellin in plant tissue culture is gibberellic acid, abscisic acid. In plant tissue culture, abscisic acid helps in promotion of distinct developmental pathway, for example, somatic embryogenesis. Ethylene. Ethylene does not find any widespread use in plant tissue culture. Stock solution of all the nutrients is prepared. Media is prepared by suitably diluting the appropriate amount of stock solutions for desirable volume of medium. Auxins are dissolved in ethanol. Cytokinins are dissolved in 0.5 normal HCl. Gibberellins are water soluble. All the ingredients are mixed, sugar is added, and finally the pH is adjusted to 5.8 to 6.0. Medium is carefully poured into culture vessels. Culture vessels are plugged with non-absorbent cotton plugs. After this, the vessels are covered with aluminium foil. For sterilizing the culture medium, it is autoclaved at a temperature of 121 degrees Celsius and at a pressure of 15 lb for 15 minutes. Finally, the autoclaved medium is stored in the cabinets in inoculation room. Third step in plant tissue culture technique is selection and surface sterilization of the explants. The explant that we use in plant tissue culture should be healthy and actively growing. Some of the commonly used explants in tissue culture are meristem, leaf, stem, flower, anther, root, nodal segments, etc. Shoot tips are used as explants for clonal propagation and for production of virus-free plants. Cotyledon, hypocotyle, stem, leaf or embryo are used as explants for callus induction. Leaf tissue is also used for isolation of protoplasts, which can be then used in protoplast culture. Pollen and ovule are used as explants for production of haploid plants. Different culture types in plant tissue culture studies are cell culture. It involves in vitro culture of cells, including single cell. Protoplast culture. In protoplast culture, protoplasts are isolated from plant tissue and then cultured in vitro. Organ culture. In organ culture, specific organ like anther, root or leaf is excised and cultured. Meristem culture. In this type of culture, meristematic tissue like shoot apical meristem or root apical meristem are cultured on a growth medium. Callus culture. In callus culture, a cell is cultured on a growth medium which forms callus. This callus develops into new plantlets. Fresh plant material is collected under water. Then the plant material is washed under running tap water with a liquid detergent like Laboline. Then a few drops of surfactant like Tween 20 is added to the plant material and again it's washed. Finally, the plant material is rinsed with double distilled water. All the subsequent steps are carried out in aseptic conditions which are provided by laminar airflow hood. Plant material is taken in a sterilized bottle and then it's treated with a sterilizing agent like sodium hypochlorite, bromine water, mercuric chloride, etc. for a suitable duration of time. Once the chemical treatment has been given, the plant material is rinsed 3 to 4 times with double distilled water. The fourth step of plant tissue culture technique is inoculation. All transfer operations are carried out under aseptic conditions. Such aseptic conditions are provided in laminar airflow hood. Laminar airflow has a motor that blows air which passes through filters that removes the particulates from the air so that an ultra clean air flows through work area. The velocity of air coming from the filters is about 27 plus or minus 3 meters per minute, which is sufficient to prevent the contamination of working area by the worker sitting in front of it. Laminar airflow has a UV lamp that is turned on about 10 to 20 minutes before being used for inoculation to sterilize the surface of the laminar airflow. In order to maintain aseptic conditions, all the culture vessels, test tubes, pipettes should be opened only in laminar airflow. It is also very important to wear apron and mask. Hands can be sterilized with alcohol. All the instruments that are used during the transfer process are flame sterilized after dipping them in alcohol. Culture vessel is opened near flame. Explant is placed into the medium and immediately the culture vessel is closed again. The next important step in plant tissue culture technique is incubation. Cultures are incubated in culture rooms where physical conditions of light, temperature and humidity are controlled. Illumination is provided by white fluorescent lamp. When light is not required, then the racks containing culture vessels are covered with black cloth. High temperature can be maintained by traditional heating systems. Cooling is accomplished through air conditioners. The relative humidity of culture room is maintained to 50 to 60%. The next step of plant tissue culture technique is plant regeneration from cultured plant tissues. When an explant is cultured on an appropriate medium containing both an auxin and a cytokinin, it can give rise to an unorganized, growing and dividing mass of cells known as callus. During callus formation, mature cells revert to meristematic state and form undifferentiated callus tissue. This phenomenon is known as dedifferentiation. Ability of component cells to form whole plant or plant organ is known as redifferentiation. different ratios of auxin to cytokinin affects the growth and morphogenesis of callus. A high auxin to cytokinin ratio helps in root development. A low auxin to cytokinin ratio helps in shoot development, whereas an intermediate auxin to cytokinin ratio helps in callus growth without there being any differentiation. There are two methods of plant regeneration. One is organogenesis and another is somatic embryogenesis. Let us first discuss organogenesis. Organogenesis is defined as production of organs either directly from an explant or from a callus culture. There are three methods of plant regeneration via organogenesis. The first two methods depend on adventitious organs arising from callus culture or directly from an explant. The third method is by axillary bud formation and growth. Now let us discuss somatic embryogenesis. In somatic embryogenesis, embryo-like structures that are formed from somatic tissue develop into new plants. There are two types of somatic embryogenesis. One is direct somatic embryogenesis, another one is indirect somatic embryogenesis. In direct somatic embryogenesis, embryo is formed directly from a cell or small group of cells without the formation of an intervening callus. In indirect somatic embryogenesis, callus is first produced from an explant. Somatic embryogenesis can be divided into two stages. The first stage is the stage of embryo initiation, in which a high concentration of 2,4-dichlorophenoxyacetic acid is used. The second stage, which is the stage of embryo production, in this stage, the medium is supplemented with no or very low levels of 2,4-dichlorophenoxyacetic acid. The next important step of plant tissue culture technique is hardening or acclimatization. Hardening is defined as preparation of the plant for natural growth environment. In vitro conditions are high in humidity, and so the plants often lack a working cuticle and stomata that saves the plant from drying out. Once we bring the plant out of culture, it needs time to adjust to natural environmental conditions. Plants are carefully removed from the media and transferred to soil or potting compost for continued growth by conventional methods. Plants are irrigated regularly with dilute nutrient solution or simple water. It's also very important to maintain high humidity conditions initially. Gradually, the plants develop cuticle and their stomata also start functioning. The final step in plant tissue culture technique is transfer of plants to the field. Hardened plants are transferred to glass or polyhouse. Plants must be irrigated frequently. The growth and variation of plants in polyhouse or glass house are monitored regularly. The hardened plants are kept in glass house or polyhouse up till the flowering stage, or They are transferred to field after 4 to 6 weeks of acclimatization. So dear students, with this we conclude today's topic of plant tissue culture technique. I hope that you understood it well. Thank you.